I'm pleased to introduce Dr. Jerry Burke.
Well, good morning. Why don't we have the panelists come up here too, Carly and Dr. Hood. I hate to hear my introduction because it's taking time out of the talking about research here, and so it's interesting. About maybe 7 years or so ago, Christy Ludlow, who used to be the chair of the Scientific Advisory Board, had what was a really interesting conference that was funded by the NIH, where she got together all of these people that were active in research or clinically very active in spasmodic dysphonia, and formed an alpha committee. And we met, I can't remember, we met at the NIH. And we were going to all sit around and discuss what research trends were important in spasmodic dysphonia. And so Christy's first question to the alpha group, as it might be the Uber group, was, how do you define spasmodic dysphonia? What are the characteristics? And the reason that she did that was that when you really want to look at a rare disorder like this, you have to identify what the phenotype is. And by phenotype, I mean, what characteristics does the individual express that tells you that this is this disease? And so we all sat around, and Christy was trying to get out of everybody, how are we going to define the phenotype? Well, it turned out that we couldn't do it. We couldn't actually come to a conclusion as to what you use to define somebody with spasmodic dysphonia, despite the fact that all these people were treating patients with Botox, and we all claimed that we knew a lot about the disease. So out of that, what I might describe as a defeat, Christy decided to run this project that Charlie alluded to, the Dystonia Project, Coalition Project. And what she decided to do was to take patients and see whether or not she could actually do a study and identify what the phenotype of patients with spasmodic dysphonia might be. And so what she really tried to do was separate patients with SD adductor from SD abductor, from patients with tremor, patients with muscular tension dysphonia. And it was a nationwide study that the NIDCD funded for that. And by the way, I just want to say that I just got off the advisory council. I had the privilege of being, the advisory council is the next level that the NIH uses to fund grants. And it was a privilege to be on it, but I can tell you that even though SD is a rare disorder, it has a very high profile at the NIDCD. And any grants that come in there are very well received by the project coordinators. And I think that has a lot to do with the NSDA keeping the spirit and the presence of SD high in the minds of the researchers. But anyway, getting back to our Uber committee, what Christy has done now, after probably 3 or 4 years of research, is she's just about ready to release a paper that will actually define the characteristics, the phenotype of patients with spasmodic dysphonia, and probably tremor, and probably muscular tension dystonia as well. So that's one of the new things that I think is important to us, because once we have a good feel for what the phenotype is, then we can go back and look at genetic problems. We can go back and look at all types of issues that might pertain to the disorder as well. The other thing that I wanted to say, that maybe a couple other new things that are off in the distance. There was I don't know if any of you know about the drug oxybate. It's a new drug that can be useful in patients with alcohol-sensitive dystonia. And there was actually a report in the literature of a patient that received one treatment of oxybate, just one treatment, and actually had a 3-year remission of their symptoms. And so I thought that was interesting. You don't know what it really means, but that certainly is something that I think we need to keep in the back of our mind. And then finally, there was a presentation recently at one of our national meetings of somebody that had treated, I think, only one patient with chronic stimulation of the RLN and actually got some relief in patients for, I think it was a 3 or 6 month period of time as well. So there's a lot of interest that's going on. And one more issue that I wanted to, Charlie talked about Kimberly Kuhlman, who actually has a fascinating grant. You know, unfortunately, the NSDA, I wish we had as much money as the NIH to study ST with, but we're able to fund pilot grants that hopefully lead to larger studies and that you're going to hear about today. But she has a very interesting study that looks at functional MRI. A functional MRI is an MRI that's done where they can actually look at, and I think you're going to talk about that a little bit as well this morning. Yeah, that actually looks at the function of the brain, the cortex of the brain, and even deeper structures, looking at glucose uptake while studying it. And at the same time, while doing transmagnetic stimulation, you can actually take a magnet that can pinpoint certain areas of the cortex and stimulate them acutely and actually study how the brain is reacting to that. And she has a very interesting study that has looked at areas that might actually be inhibitory to other speech areas and that there may be deficiencies in this inhibitory area of the brain, and so the NSDA has decided to fund that study. It's a very exciting study and very original and innovative. In addition to that, Dr. Patel's another pilot study that we're looking at is looking at high-speed dynamics of the vocal cords as they vibrate and go together as a way of kind of identifying more clearly what types of processes are contributing to the dystonia that patients experience when they try to speak with spasmodic dysphonia. So, with that brief introduction about all the exciting things that we're doing and looking at, I'd first like to introduce Dr. Tanner, who's going to talk to us a little bit about her research in the epidemiology of spasmodic dysphonia. Thank you so much.
So I am a neurologist, and I also do research in epidemiology. I work in the area of movement disorders. So one of the disorders that comes up for us is dystonia, and I've recently been working in the question of looking at the epidemiology of the primary focal dystonias, including laryngeal dystonia. So what's epidemiology? Epidemiology is looking at the distribution of disease in populations. So, you know, in San Francisco, how many people have a particular problem? And this is important because if you know everybody in the population, it gives you information not just for health planning, and it can also lead to clues for the causes of the disease, can help us figure out what we need to do, and it can also help us think about what we call the determinants, so causes, protective factors, risk factors, or evaluate treatments. And so this is an important area of research, but there's been relatively little done in this area, and part of the reason for that is because dystonia and laryngeal dystonia in a big population sense is relatively rare, and yet, as you just heard, figuring out who's got it is not all that easy, right? So somehow an expert has to be involved in this to figure out, is this disorder or is this something else? And is it one disorder, or is it, you know, in the context of people with more dystonic features, figuring out the difference between voice tremor, muscle tension dystonia, it can be challenging. And then finally, to understand scientifically the determinants of disease, as neurologists and neuroscientists, studying the brain after someone's passed away can give us lots of useful information about the chemistry or the anatomy, the areas affected, and that's really not been possible with disorders like spasmodic dysphonia or laryngeal dystonia. So this is a little bit about the background of what's been known prior to the work that I'm going to present that we've been doing in Northern California. And what we know is that it appears that there's a Caucasian and female preponderance, that age at onset is about 45. But the problem is that this information pretty much comes from clinics. And so It only is based on who went to the clinic, and it's possible that, there are other people or there's a different pattern, and they just weren't attending the clinics where this was counted. So important starting point, but not necessarily the final answer. So prevalence means how many people are there today in a given area who have a particular problem. And so as you can see, just from looking at the bars here, there's not a lot of agreement about how many people there are in these studies that have been looked at. This is Poland. This is a bunch of European nations together. Poland, Munich, Belgrade, Oslo, Northern California, UK, and Rochester. So this is our study. And it ranges from about 52 to about 3 per million people. So quite a wide range of variation. Is there agreement about whether it's more common in women or not? Well, maybe a little bit. This again are from clinical series, and it looks like certainly in the UK there appears to be a female preponderance, but when you get down to Munich it flips. In Rochester, Minnesota it seems to be about equal. So again, we don't really know these answers, and more work needs to be done. The reason this is important is it gives us clues to what might be determinants of the disease. So that's why we're really focusing on it. And again, the problem is that these are what we call prevalent cases. It may not represent the entire picture of the disease. It's the people you can count, and it's also people who have had the longest survival with the disease. So it may not be just who has the problem, but who has it and lives a really long time with it. So it's not necessarily the complete answer. So the way to get around this is to look at what we call incident cases. So incident cases in an epidemiology way of thinking are the number of new cases in a defined population for a period of time. So that gives you a better idea of the sort of determinants of disease. The dilemma is that until the work I'm going to present to you, there's been exactly one incident study of spasmodic dysphonia. Jay Nutt did this. He published it in 1988. He looked in Rochester, Minnesota, in the Olmsted County Mayo Clinic population, where they can look in the whole county. The thing is, if you'll notice, he looked over a whole 30-year period, 1950 to 1982, and in that time there were only 4 cases identified. So it's a small population, 4 people over 30 years. Doesn't give you a big insight into exactly what's going on. So we felt it was time to do more work looking at all of the primary dystonias. And so we chose Northern California Kaiser. And the reason we did that is that in Northern California Kaiser, the number of people who are members there are about 3 million at any given point in time. And because it's a health plan, you always know exactly how many people and who are the people in Kaiser. So you know, are they male, are they female, how old are they, where do they live, all that stuff. And so you have what we call a defined denominator, and that's important for making your estimates. It's a big enough population that we have a chance of really understanding some things. And in Northern California, it's pretty much reflective of the whole population of Northern California. About 1/3 of the people here get their health care at Kaiser, and in general, they're pretty similar to the rest of the population in Northern California who live here. So it's a good place to look for disease. It's a good study laboratory for us as epidemiologists. So what we did is we looked for all the primary dystonias. We started out with about 8,000 names of people who had been diagnosed. We spent a lot of time figuring out how to find them. I won't belabor that, but I can talk to you about it if you're curious. And then we were looking for new cases within this period, 2003 to 2007. I and my colleagues did an expert review on this. We used movement disorder neurologists, and then also for laryngeal dystonia, collected information on whether or not they'd had laryngoscopic diagnostic studies. And for spasmodic dysphonia, we found 140 individuals who had spasmodic dysphonia. 126 of those seemed to be primarily spasmodic dysphonia or focal, and then 14 had other features. So where this left us in terms of description, we had about 81% women, about 19% men. Average age at diagnosis was about 55. 80% were Caucasian, sort of similar number of Asian and African American, 2% Native American, 1%. So this is just describing the numbers. We'll look at how that relates to the population in a minute. And we asked people to self-identify, all these are self-identified, but as Hispanic or whether or not people were Ashkenazi Jewish, because some forms of dystonia are more commonly inherited in that population. And only a small number of people actually responded to those questions, but we did get that information as well. For the SD population, 96% of them had been seen by a specialist and 80% of them had a laryngoscopic diagnosis. So a fairly high level of diagnostic certainty here. And this is what we found. So these are other forms of focal dystonia: cervical dystonia, blepharospasm, involuntary eye closure, involuntary neck turning. And the third most common of the focal dystonias was laryngeal dystonia or spasmodic dysphonia. So fairly common in the context of primary dystonias, more than people might have thought, I think, beforehand. This just shows, looking by gender, so adjusting for the numbers of men and the numbers of women in the underlying population to avoid skewing, looking at the pattern over years of age. So this is less than 20, this is 80, and as you can see, this big green line is laryngeal dystonia. It kind of goes up with increasing age, and then maybe becomes a little bit less common, not prevalence, but incidence, so number of new cases later in life. Looking at it within gender, and this is taking into account the denominator, all the people who are male or female in the underlying population, we found a female preponderance of laryngeal dystonia. So this is women, this is men. You can see it's more common in women. And looking by distribution across racial groups, again, now adjusting for the number of people in different racial groups in the whole population, we found Caucasian to be the most common group. This is Asian, this is African American, this is Native American. The only caveat to this, what looks like a real excess in Native Americans, we only had really a few cases. So even though we adjusted it, I think precision is very bad for this. So something to explore further, but I wouldn't take that as solidly as the Caucasian number. We also looked at things that might be triggers or risk factors. Other people have reported this, and what they've reported in the past are that if they're comparing people with spasmodic dysphonia to normal controls, things like infections, hospitalization, surgery, trauma, injuries, or family history of neurologic disease were more commonly associated with laryngeal dystonia. If they compared relatives to people with related but with spasmodic dysphonia, they found that there's certain infections or maybe stress or upper respiratory infections might be risk factors. And then if they compared to other voice disorders, what they found, again, this idea of infection or people who are using their voice frequently in different situations might be risk factors. So these are different studies from clinic series, and again, getting out of the clinic and looking in the population can sometimes give you a different picture that could be more accurate for all cases of the disease. So what we did is that we looked in two ways. First, we took people that we found in this Kaiser population who had laryngeal dystonia, and we matched them. So we found people who didn't have it, were pulled out of the population, but were the same age and the same gender. And we asked them questions and talked to them on the telephone to do interviews about things like medical history, lifestyle, all the things we talked about there. And what we found in this population, comparing people with SD to similar controls, was that people who had ever held a job or had a hobby that required repetitive use of the voice was more commonly reported in people with laryngeal dystonia than people without. So kind of what we call excessive or overuse seem to have some relationship. The odds ratio is a measure that epidemiologists use, and it says basically if you have a job or a hobby that, like singing or, you know, phone operators or something, that you use your voice a lot, you're about 8 times more likely to come up with SD. So there's some factor that has to do with overuse that seemed to be important. Interestingly, we also ask about smoking. This is important in some of the other neurologic diseases we look at. So we always ask about these lifestyle behaviors. And we found that people who were smokers were less likely to have SD, right? And we've seen the same, it's funny, I know, and it doesn't mean you should go smoke, right? But we've seen the same pattern, for example, in Parkinson's disease, and there we've been exploring it quite a bit, and what we think is it relates to perhaps some of the brain chemical messengers that can be affected in Parkinson's, and we actually have a trial right now where we're giving nicotine patch to people with early Parkinson's to see if it slows progression. So, you know, there's maybe some plausibility here. Again, these are clues that we can get from looking in populations. They may be wrong, but it gives us something to try to investigate in the laboratory or maybe in human populations. So the caution here is that a lot of these things could be subject to what we call bias. And bias means, you know, it looks like it's real, but it's not because there's some other factor that's determining it. For example, what we call recall bias is one example. If you have a problem, you might spend a lot more time thinking about how did this happen to me and what's my past history. And we call up a control and say, can we ask you some questions? And they've never really, you know, they're fine. They never thought about this stuff. So they might not give such a complete answer. So those kinds of things can affect this. So we took another approach in the same population to try to control for that idea of bias. And in this case, what we did was we used information that was collected just by virtue of people being, you know, getting care in Kaiser that we could get out of their medical record. So we looked at people with laryngeal dystonia, and here we could match a single case to 10 controls. So we could be really sure, you know, that there was a difference between the people with dystonia and the control. And before we started, we said, you know, we have some ideas of questions we want to explore based on what other people have reported. And so we looked for what we call diagnostic codes, people who had been diagnosed with different kinds of features prior to the diagnosis of dystonia in the person with SD or prior to that same equivalent time point in the controls. And what we found was that people had been diagnosed with throat infection, primarily strep throat, prior to the time of their diagnosis, more commonly if they went on to have spasmodic dysphonia than controls. And here it was almost a 6-fold increased risk. Now, this is an interesting thing to explore because on the one hand, maybe this is a risk factor, and in fact, some of those case control studies had a similar finding that throat infections were a risk factor for SD in their clinic populations. Or is it just simply people went in with some changes in their voice and it was a diagnostic error, right? So they didn't really have a throat infection, but that's what the primary care physician's first take was, oh, you got a sore throat, your voice a little hoarse, that's what it is. So it's a question that needs research. The fact that it was called strep throat and that some of it had been diagnosed leads me to believe it wasn't completely that diagnostic error because that requires a laboratory study. But it's still a question that needs further investigation. We also looked for family history and looked for genotype changes in certain genes that had been associated with other primary dystonias. We had relatively few people who had a family history of dystonia, but we had about 5 cases who had affected parents, or 7% of the people who responded. Not everyone did respond. Spasmodic dysphonia was identified in 2 of those cases. So a suggestion of a genetic basis in some people, probably a small effect, but if you can identify a family and study them closely, sometimes that can give you a lot of really important information that could extend to causes in other people without a family history. The other thing we looked at, and this was touched on a little bit in the beginning presentation as well, was the whole idea of diagnostic error or diagnostic delay. It's a common thing in all of the primary dystonias that people are not believed to have a real disease, and they need to go from doctor to doctor to doctor till they finally get the right diagnosis. So we couldn't look at error, who has the disease, was never diagnosed, because we weren't examining everybody in the population. But we could look at delay, which is kind of a way to get to that. How long did it take you from the time you first presented with a symptom to the time you got the right diagnosis? And what we looked at, the same way, survey and then looking backwards in everybody in the electronic medical record, we found that the time from the onset of symptoms to the correct diagnosis was on average somewhere around 5 years. Looking at, there were some people who was really long, really short. So another measure, looking at the median, which kind of adjusts for those outliers, it was still about 2 years, right? So a pretty long time from presenting with a problem to getting the right diagnosis. On average, about 4.5 doctor visits till getting the right diagnosis. And then we asked people to respond to a survey about how this affected them. And 65% of people who had a diagnostic delay reported that this affected them adversely. And the kinds of adverse effects were worry and concern about themselves and their well-being in almost everyone, 94%. 79%, almost 80% had problems at their job because of this. It affected their employment. 64% it affected their family life and caused stress in the family. So really significant problems related to this. So just to summarize, where does this leave us? I think case definition, case recognition, education of the community and the healthcare community, as well as the general community, about laryngeal dystonia is very important, so we don't have these long delays in diagnosis. Figuring out more from these clues about risk factors, this can help us perhaps come up with ways to delay, prevent, or understand better to even get rid of the whole disease. That would be our long-term goal. And then taking these back to the laboratory, doing modeling in the laboratory to understand more about it can lead us maybe to better treatments or, again, prevention of the disease. And then I'd like to just stop by thanking, this was a study that involved lots and lots of people, so my many colleagues working in this project, as well as all the people with spasmodic dysphonia and their families who participated in the research. Without their involvement, we'd never get anywhere. So thank you very much.
Thank you, Carly. That was extremely illuminating. I think it's interesting that spasmodic dysphonia really cuts across all socioeconomic categories. And in fact, there are very famous families in the United States that have held tremendous positions, President of the United States, where if you listen to their family, you recognize that spasmodic dysphonia has been an element in their family for very many years. Christy used to always say that she felt as though there was a genetic predisposition and that there was oftentimes a triggering event that led to the development of the disorder. And as we understand the phenotype better and the genotype better, maybe we'll finally figure that out. Next, I'd like to present Dr. Hood, who studies the effect of sensory changes on the development of the disease and the way the disease manifests itself. And apparently he has a brand new study and an experiment that he's working on that he wants to present to us this morning as well. Thank you.
All right. Okay. Hi, I'm John Hood, and today I'm going to tell you about a study that I'm beginning in collaboration with Sri Nagarajan and Mark Corey, trying to look at what causes spasmodic dysphonia by looking at the timing of neural activity in speaking in spasmodic dysphonia. So in my lab, I study how the central nervous system, or the CNS, controls speaking. And in particular, our lab has had years of experience studying the role of sensory feedback in the process. And this is particularly relevant to spasmodic dysphonia because, as Mark said earlier, spasmodic dysphonia is likely a disorder of the CNS. Some of the circumstantial evidence for this is that SD is task-specific. It occurs in speaking, not laughing or crying, even though all three of these vocal behaviors use the same larynx. And in its task specificity, SD resembles another neurologic disorder called focal hand dystonia, or better known as writer's cramp. And what's significant about this is that focal hand dystonia, studies have shown, is associated with abnormal somatosensory representations and has been theorized to result from a runaway feedback process of the sensory motor loops governing hand movement. There is also some limited evidence that sensory feedback alterations, like speaking while hearing masking noise, can improve SD symptoms. And this has led to the idea that SD may arise from abnormal CNS responses to sensory feedback during phonation. This fits well with a model we've developed in my lab for how the central nervous system controls phonation. And in this model, production of an utterance that involves phonation begins with higher frontal cortex, or the inferior frontal gyrus, activating what we call a phonation control network consisting of motor cortex, somatosensory cortex, and auditory cortex. And during phonation, in motor cortex, ventral premotor cortex maintains a running estimate of the current dynamic state of the larynx that primary motor cortex uses to generate controls of the larynx. And while the larynx is responding to these applied controls, the ventral premotor cortex receives a copy of these controls, so-called efference copy, which it uses to predict the next laryngeal state. Meanwhile, the larynx is responding to these controls and generating somatosensory feedback and auditory feedback. And to process this incoming feedback, ventral premotor cortex feeds forward the laryngeal state estimate to the higher sensory areas, as shown in the green path here, that they use to predict the incoming feedback. So then, at the primary sensory cortices, the incoming feedback is compared with the predicted feedback, generating feedback prediction errors. And these feedback prediction errors are then fed back to ventral premotor cortex and used to correct the estimate of the laryngeal state, which in turn is fed back to primary motor cortex to generate further laryngeal controls. So the upshot of this is that during phonation, there are loops of neural activity between the sensory cortices and motor cortex. And by analogy with what's been theorized for focal hand dystonia, this has led to a hypothesis that abnormally large neural responses at any point in these sensory motor loops during phonation could lead to a runaway feedback condition resulting in the strained vocal behavior seen in SD. So this hypothesis formed part of the motivation for this study. We're about to begin examining the timing of neural activity during phonation in SD. Another motivation is that in steady phonation, with a lot of areas all activated and connected to each other, it's difficult to identify the initial cause of a runaway feedback situation, if you're following that hypothesis. So to identify such potential causes, we propose to use magnetoencephalography, otherwise known as MEG, and laryngeal electromyography, or EMG, to examine the timing of neural activity in SD surrounding two events in phonation. Phonation onset and what I'll describe further as pitch feedback perturbation. So what is MEG? MEG is based on the idea that neural activity in cortex generates magnetic fields that can be measured outside the head. And if you have enough magnetic field detectors arrayed around the head, you can localize when and where neural activity occurs in the brain. So just as an example of a picture from some analysis from MEG, here's results of MEG study of responses to pitch feedback perturbation showing activation in left auditory and parietal areas 100 milliseconds after the pitch feedback perturbation. And then here's a picture of the evolution of that activity at 200 milliseconds after the pitch feedback perturbation. So MEG is similar in many respects to functional MRI, except that it has a highly sensitive millisecond scale time resolution on resolving neural activity. So this is our setup for the experiment, with showing the subject with his head in the MEG detector array, wearing earphones and a microphone, with a DSP being used to perturb pitch feedback that the subject hears in their ongoing phonation, and a PC being used to orchestrate the whole experiment and to visually prompt the subject when to phonate. What's not shown in this figure is that we will also be monitoring laryngeal muscular activation using EMG based on hook wire electrodes. And here's a picture of a subject in our MEG scanner participating in a similar experiment. So a typical trial of the experiment begins with the computer LCD screen displaying disappearing dots that prompt the subject to begin phonating the vowel, steady phonation of the vowel ah. And sometime during the phonation, the ongoing phonation, the DSP is directed to alter the pitch that the subject hears of the feedback of their ongoing phonation. The trial ends with the reappearance of text on the screen. And then this trial diagram allows me to point out the two events that are critical to the analysis of the experiment. First is phonation onset, and then second is onset of the pitch feedback perturbation. And the critical thing about this study is that MEG allows us to examine the neural activity of cortical areas in the times around these different events. And that timing information allows us to determine the functional significance of any abnormalities we see in activation in spasmodic dysphonic patients. To see what I mean, consider the neural activity at phonation onset. So At phonation onset, after the subject is prompted to begin phonation, it begins with motor cortex sending laryngeal controls to the larynx, and at the same time, somatosensory predictions being generated in somatosensory cortex. So if we use laryngeal EMG to monitor muscular activity, we can detect when the larynx begins to move in response to these applied controls, and use this to discern to discern functional significance, because any activity that happens before this point would have any abnormalities in neural activity seen before this point would have to do with the feed-forward process of generating laryngeal controls and generating somatosensory feedback predictions. Whereas any abnormal activity seen after this event would have to do with response to somatosensory feedback, which could occur in somatosensory cortex or downstream in motor cortex. We will also be monitoring, of course, audio sound output from the subject, which we can use to detect the onset of phonation that occurs subsequent to laryngeal movement. And any abnormal activation that we see after this event would have to do with responses to the somatosensory or auditory feedback responses associated with onset of phonation. To isolate specifically whether there are abnormalities in responses only to auditory feedback, we use the DSP, the digital signal processor, to induce perturbations to the pitch of how the subject hears their ongoing phonation during the trial, which this pitch feedback perturbation produces auditory feedback prediction errors in auditory cortex. And so any activity seen downstream after the pitch feedback perturbation would be activity associated specifically with the response to auditory feedback. So to sum up, this experiment is generally a 2-hour experiment in the MEG scanner, which will run on patients with SD and age-matched controls, comparing results between the groups, as I've just described in these past diagrams, to isolate abnormalities associated with spasmodic dysphonia. And the results will allow us to determine what areas of the CNS hopefully function abnormally in spasmodic dysphonia, which, as Mark alluded to earlier, should allow us to develop focused treatments that are directed specifically to those areas. So, thank you. And just one more. If you're interested in being a subject, you can contact the UCSF Voice and Swallowing Center at the number shown here.
Thank you, Dr. Hood. I asked Kim whether we had time for questions, and she said 1. So, might there be a question from the audience? Okay.
What sort of treatments are being proposed that would target a section of the brain? Are these drugs or are they?
Well, Hello? Oh, yes, this is on. Oh, what sort of treatments have been, the question was, what sort of treatments have been proposed to specifically target abnormally activating areas in the CNS? Well, one possibility that we're considering pursuing in our lab is the use of repetitive transcranial magnetic stimulation to specifically target and reduce the reactivity of specific regions, areas in cortex. Repetitive TMS, transcranial magnetic stimulation, is a technique for, it's kind of the opposite of MEG. It's imposing magnetic fields on the brain to stimulate neural activity in the brain. And repetitively doing that can actually decrease the reactivity of the area being treated with TMS. And such decreased reactivity could lead to reduced runaway feedback situations that may lead to spasmodic dysphonia. So that's just one example that we're considering pursuing.
Okay, thank you very, very much.